Hello, my name is Victoria Sarver, and this is my senior research thesis on butterflies of Saint Vincent College diversity across campus. So, to begin, this project will look at butterfly diversity and how to increase it. In doing this project, I hope to learn what the diversity across St. Vincent College campus looks like and if we can improve upon this to help our environment. There are many suggested methods on how to increase butterfly presence and diversity, but this project will specifically look at food sources. So this is done by adding flora to the trap sites that I have set up. There's about 6 of them across campus. So my hypothesis is that this treatment will in fact make a difference on the number of butterflies and the diversity of what is caught here on campus. So my methods. So as I said, I set up 6 traps across St. Vincent College, and these are the various locations. So for the first 3 weeks, these traps were set up in the morning and were left out for 24 hours before the butterflies were collected. For the second 3 weeks, there were a number of different flora placed out so that the butterflies would be enticed to come and try to get trapped. So trap setup. So the traps were set on these garden hooks that had another length at the bottom to make sure that the traps were above the ground. So after the traps were popped up, nectar was added to the dish underneath to get the butterflies to fly into the trap. Along with this, I also had an online source that collected the average temperature, cloud coverage, wind speed, and precipitation of the area during this time. Finally, at the end of the 24 hours, the insects and butterflies were counted and the species was noted, and all of them were released back into the environment. Like I said before, the treatment was added after the first 3 weeks. So on the 4th week, the 2 species, Monarda didium and Asclepias tuberosa, were added to all of the 6 sites. So 1 of each plant was added. But unfortunately, the plants were not thriving in these conditions, and so a 3rd plant was added, the Rudbeckia hirta, to each site. So there was 2 of those at each site, making a total of 4 plants by the 5th week. So now the percentage of each type of insect was found over the total catalog that I did during the experiment. Data about each area was also taken, as these can affect butterfly presence. So each area we looked at shade, the nearby flowers that were already there, and the relative closeness to buildings nearby. As I said before, weather data was collected for each day, and then comparison between before and after treatment was also done as part of data analysis. So now on to our results. As you can see, we captured a number of different pollinators. So of the 106 insects captured, 25% of them were the peckius. And so this was the only butterfly species that was collected during this experiment. And the other species have a range of common names such as bees, flies, and moths. These 3 tables are looking at the data observed from the areas nearby. Table 2 is looking at the shade, Table 3 is looking at flowers nearby, and Table 4 is looking at relative closeness to buildings. For Table 2, the observed versus the expected are relatively similar with the chi-square analysis. The chi-square number for this table was 0.57. But for Table 3 and 4, the observed versus the expected are different. For Table 3, the chi-square value was 21.03, and for Table 4, the value was 23.58. All of these were at a DF of 1 and with a significance level of 0.05. These next few graphs are looking at the daily weather. So these 2 graphs cover the cloud coverage. So cloud coverage was described in 5 different ways: either fair, partially cloudy, mostly cloudy, cloudy, or precipitation present. These numbers were then given values, and the average was found for the day. So this average was then compared to the daily number of peck skippers and total pollinators collected. As you can see, the peck skippers have a negative correlation and the pollinators have a slight positive correlation. But both of the R values are low, meaning that the graph does not describe a lot of the variability in the data. Next, we are looking at precipitation. For both peck skippers and pollinators, there is a positive correlation, but again, that R-square value is fairly low, meaning that the data is not properly represented. These two graphs then look at daily temperature. So the average temperature for each day was found, and that was compared to the numbers. So there is a positive correlation for both peck skippers and total pollinators, but both of the R values are again fairly low. Finally, we have wind speed. The average wind speed for the day was found and compared to the numbers. So for peck skippers, there is a slight negative correlation, but for all pollinators, there is a positive correlation. But again, both of these have an R-squared value that is relatively low. Finally, we are looking at treatment before and after. As I mentioned before, the treatment before initially was nothing being present and just the traps for the butterflies to go into. And this was done on the first 5 days. The last 6 days had the treatment of the 3 different kinds of flora present underneath the traps. So as you can see, for all pollinators in green, there is a slight decrease, but for peck skippers in blue, there is a slight increase. So now, what does all of this mean? At the beginning of this experiment, I hypothesized that there would be evident butterfly diversity across campus, and I would be able to increase this with the influx of flora and plant available to them. But unfortunately, this does not seem to be the case. So from the data, it can be concluded that Saint Vincent College does not have a diverse butterfly population. Of all the pollinators that would cross over the course of this experiment, only one was a butterfly species. And as I said before, this butterfly species was the peckius, or peck skipper. So there is also not enough evidence to conclude that shade and the number of pollinators is associated. This is based on Table 2 and the p-value being greater than the significance level. But according to previous research, there should be more butterflies in sunnier areas, as this is an environment that they like to be in. Next, flowers and being near buildings do have some sort of effect on the number of pollinators because both of their p-values are less than the significance level. And this was Table 2 and Table 3. The flower data agrees with previous studies. As flowers increase, the population around them should increase, and this does not only include butterflies, but also other species to get a diverse population. And buildings affecting butterflies also agrees with previous research, as the area around the butterfly is urbanized and there's just more activity, there's going to be a decrease in population and diversity. So when looking at weather, a few things can be concluded. When looking at cloud coverage and peck skippers, there is a negative correlation. And for other things, there are also negative correlations and positive correlations. But due to those R values being relatively low, only a certain percentage of the variability is accounted for within this data, meaning that the correlations are low, and there isn't very strong statistical evidence that any of these correlations are right. So next on to the floral treatment. So as I said, after the 6 days, those florals were added, and the total number of peck skippers did increase, but the total number of pollinators did not. So this can mean that the treatment was not as effective, but also the diversity was not increased, which was another part of the hypothesis. So again, it can be concluded that we did not get the effects that we were hoping for. So finally, this experiment was successful. I did learn that the main type of butterfly species on St. Vincent campus was the pollitis peckius, or peck skipper. But in the future, experiments may want to collect data for a longer period of time. Another experiment showed that after a 12-year period, they were able to see an increase in butterfly population and diversity from having increased flora. But another critique of this experiment might be the amount of flora that was chosen. So at the beginning, the flora came in smaller than anticipated, so maybe more flora needs to be added to be more enticing to butterfly species. Other factors can also be collected during experiments, such as how often do people walk through these places, or what the elevation is of where the traps are set up, because both of these things also correlate with butterfly diversity and population and how often they're going to these areas. But overall, there's still a lot more to learn about butterflies on Saint Vincent College's campus, and future experiments are needed for this research. So I would like to thank Dr. Kellum, as he was my advisor during this project and helped me along the way. I would also like to thank the Biological Sciences Department for the last 4 years and for my education. And lastly, I would like to thank the Palumbo Grant Endowment, as they were able to fund my project and give me the resources that I needed to be able to do this experiment. Here are all my works cited, and thank you for listening.